with something wrong, either have an extended stay or leave with something different than what they came in with. All right. Some examples are C. diff, E. coli, norovirus, Pseudomonas. A lot of these infections are caused by modern medical procedures and just exposure, extended stays in hospitals. We'll talk about these in just a second. MRSA is becoming a common or more common infection that people can acquire when visiting the hospital, right? And a lot of this, right, has led to, we'll say, the refinement and the recommitment to procedures that workers and staff and healthcare providers are taking as they interact with patients. And you recognize this name, Ignaz Semmelweis and Oliver Wendell Holmes. Like Ignaz was a big supporter of hand washing before it was popular. And again, this was a really important health measure at the time and continues to be one that, interestingly enough, sometimes is neglected still. All right. So this is just a kind of a short little video from your textbook.
Nosocomial infections are those infections acquired by patients while they're in a healthcare setting. It's estimated that 30% of nosocomial infections are preventable through more stringent use of aseptic technique by healthcare personnel. There are 5 major categories of nosocomial infections. The most common type of nosocomial infection is a urinary tract infection. These are typically contracted during insertion or removal of urinary catheters, and usually involve the normal flora of the patient or healthcare worker. The second most common type of nosocomial infection is a surgical wound infection, again caused by normal skin flora that get into the surgical wound. Lower respiratory infections are usually associated with respiratory devices. Cutaneous infections commonly occur in newborns and burn patients. Bacteremias are another type of nosocomial infection, usually due to intravenous catheterizations or injections. All other infection types account for about 11% of all nosocomial infections. Patients are susceptible to nosocomial infections because their immune systems are already weakened. The stress of surgery on the body also makes it harder to mount an adequate defense against these microbes. The exposure of the compromised patient to antibiotics, whether as treatment or as aerosols in the environment, allows resistant microbes to become abundant and cause disease. Healthcare personnel are often carriers of resistant microbes. The need for invasive procedures such as injections, insertion of catheters, or surgery increases the chance that microbes from the skin can enter the bloodstream. And finally, the constant movement of staff and visitors among the patients increases the chance that microorganisms can be passed from one person to another.
So what's really interesting, and they kind of pointed it out, is that patients in a hospital, they're getting treated, right? And the very same thing that is treating them and helping them heal could also be making certain pathogens more resistant, right? So it's really challenging. And I mentioned surgical wounds. Well, I know for a fact that a lot of hospitals are trying to be really careful about how much antibiotics that is prescribed to somebody after surgery, right? And antibiotics are good after surgery because they prevent infection, but overuse of antibiotics leads to resistant organisms. And so it's just kind of a really interesting balance that we're currently trying to figure out, right? And so here, this is just showing you a couple of different types of those infections, right? So exogenous, right, pathogen acquired from the healthcare environment. Right, so that got mentioned there. Endogenous, right, pathogens arise from normal microbiota within the patient, right? So they're talking about the catheter, right? This could also be some GI issues, all right? Latogenic, right, results from modern medical procedures. So that could be some types of surgery, right? A lot of different things there, and super infections, right? So you have these antimicrobial drugs that basically destroy your normal flora, and then you allow other microbes to survive, right? So this, an example of this would be C. diff, right? And so a patient comes in, they're really ill, you give them antibiotics, they start to do better, but those antibiotics wiped out all their good gut bacteria, and then the bacteria Clostridium difficile has an environment that they can move in on, right, and then cause an infection in that patient, right? And I don't think it's surprising. I know a lot of you have worked in a healthcare setting. You probably agree that, again, the worst place that you could catch an infection is in a hospital or a healthcare facility, because if you've ever had to clean up anything there, right, the chemicals and the antiseptics that they use are really, really powerful. And so if something survives that, it is definitely not an infection that you want. It also kind of points out how delicate the balance is. I really want to pick on Abby again, but I'll go after Caitlin today. Right, and that's hard, right? So Caitlin, let's say she's cleaning up something in a healthcare setting, and she does her job 99%, efficacy. I mean, she's really good. Like 99% on her test, she'd be really, really happy with that. I assume, right? Okay. She's like, no, I want to write. So that a little bit that she misses, right, could then end up causing an issue in the patient. And that's not really a knock on, people that are cleaning things up. I mean, people miss things, and it's not even on purpose, but the system is really delicate, and it doesn't take much for someone to potentially get an infection in a healthcare setting. All right, and I do like this Venn diagram, right, from your book, and it's just showing there's a lot of different things that contribute to the hospital-acquired infections, right? You have immunocompromised patients. If you're in the hospital, you're probably not just hanging out there getting treated, sleeping overnight because you're healthy, right? You already have things going wrong. There are microorganisms that are present in the environment, right? In the hospital room, right? They mentioned in the video, healthcare workers are carriers, right? And then the transmission of things between staff and patients and among patients, visitors in and out. I almost guarantee that a number of you probably have MRSA on your skin, and it's just kind of chilling there. There's no issues unless you get some major abrasion, right, in your skin. A lot of us are carriers, and so just looking for that opportunity. Sometimes these settings are great opportunities. All right. I have an answer.
I'm kind of confused by what you mean by like we're carriers, but we don't. Like, I mean, like it's just on us.
Yeah. So, right, we talked about your kind of two major barriers, right? Your skin and your mucosal membranes, and so you can have resident pathogens that are hanging out on your skin, but your skin is a great barrier. So they're basically they're living there, right? They're part of your normal flora, and it's not until you have a disruption or an injury to that protective layer that they can move in, right? And so we see soccer players actually a lot of times in AstroTurf, right? Because they're like a lot of people move to the artificial turf, and they go and slide, and they're more prone to open up their skin now, like basically a hardcore rubber, and then slide on an AstroTurf. So we've seen increases in MRSA infection, in them. So it's, again, that's why they're opportunistic, right? And again, different people's immune systems can handle things differently. So you're super, super healthy, right? You're a carrier, and then you transfer that to somebody who's immunocompromised, and they can't control it like you could. Does that help a little bit? You're welcome. All right, so let's talk about innate immunity. And, some of this kind of conveniently plays into Alanis's question about how we can control, recognize, and react to things. I promise I did not plant that question. All right. But what we're going to talk about moving forward here today and a little bit on Monday is about the cells that are involved in the innate immune system, like get a basic understanding of who they are and where they're from. Understanding how these cells, along with certain barriers in your body, work and contribute to the concept of innate immunity, right? And then one of the most important things for me, right, is showing you or starting to introduce the idea of how the innate immune system and the adaptive immune system interact, right? They get presented as separate units. I think it makes that a little easier to digest. But truth be told, they rely very, very heavily upon one another. All right? So this is, right, this is Ila Mechnikov. He won the Nobel Prize, you can see here, in 1908. And he, right, is credited essentially with discovering the innate immune system. And what he would do here, you see his quote, but he would take little thorns and he would stick them into starfish larvae. If you're wondering that's what a starfish larva looks like, I've seen grown starfish, have you? Cool. Right? But he would stick these thorns in, and then he could observe these cells kind of rushing to the thorn, right? Responding to that injury, right? And responding to likely, you probably did not really realize it at the time, but bacteria that's probably on that thorn that's causing an issue, right? And so those cells, right, that were rushing to that site of injury, right, this was our first indication of the innate immune system. So before we even get to the cells, right, let's talk about the basic premise of innate immunity. It is the first line of defense, right? So we've talked about your mucosal barriers, we've talked about your skin barriers. The idea being that if those barriers are effective in doing their job, they prevent the pathogen from even getting into your body, right? Now, we've all been sick, so we know that's not a perfect system. And so once those pathogens get in, right, the innate immune system is charged with detecting them, all right? And we'll talk about that in a second. The detection can be responding, right, to that pathogen itself, or that detection can involve alerting other parts of the immune system, aka the adaptive immune system, which will be our next kind of unit or section that we look at, right? And then once identified, right, they will try to destroy that pathogen, right, or that threat. Again, some cases they'll be able to handle that pathogen or that threat on their own. In other cases, they'll have to pull in the adaptive immune system. So we'll start to talk about that interplay. All right. So this is a diagram from the old textbook that I used to use. And I kind of like it. It treats the innate immune system like a fort, right? That you're kind of hunkered down in. And so you have your security walls, right? The first line of defense, so your skin and mucous membranes. We'll talk about this a little bit. There are also antimicrobial substances that are secreted onto or into these membranes, right? One of the things I don't think we appreciate is your skin is really salty. Have you guys done, no, you haven't done MSA plates in the lab yet, have you? Yeah, I don't think so. It's probably a little early for that. But that's a mannitol salt agar, right? And in that media that you're growing bacteria on has a really high salt concentration, right? A lot of bacteria don't do well on high salt concentration. So your skin, right, if you were to lick it, which please don't do that right here, all right, until you're out of my classroom, it would be really salty. And one of the reasons your skin is salty is it actually controls what pathogens, right, are present or what normal flora is even present on your skin, right? And you can secrete things into your mucous membrane too, and I'll talk about that a little bit later. So again, our first line of defense, detection, right? Maybe they've gotten by that first line of defense, so what do? Well, you have something called pattern recognition receptors. We'll talk about a couple specific examples of those. They are looking for patterns, right, molecular patterns of pathogens. So their basic job is to say, this is self or this is non-self, all right? And in this case, no matter what the non-self is, the reaction is going to be the same. So if you've studied the innate immune system before, maybe you've heard it called the non-specific, which I think is not quite fair, but that's where you kind of get this name, right? They, or the adaptive, or the innate immune system is not distinguishing between specific pathogens necessarily, although I'll show you kind of exceptions to that. They are just saying, this is self, this is non-self, we have to go after it, right? We have something called the complement system, which we'll talk about a little bit later, that is great at removing pathogens, specifically bacterial pathogens. And then once, right, we have identified and we want to destroy, right, we have something called an interferon response, which hopefully you remember from the quiz, phagocytosis. Morgan, what's phagocytosis? What does that mean? Okay, maybe a little more specific. It's true, you destroy stuff. Things do to get destroyed. What's phagocytosis? Huh? Eating, right, still eating, right, phagocytize, right, they ingest the pathogen. Complement activation, right, inflammatory response fever, which we'll talk about all of these in just a second. Different things that your body is doing to try to destroy pathogens. Right? So the role of skin, right? For your skin, right, you have two major layers, the epidermis and the dermis. The epidermis, right, is made of something called stratified squamous cells. So you basically have these layers of cells that are stacked on top of each other, right? The part of your skin that you touch is actually dead cells, right? So you're basically covered in dead stuff to protect you, right, from pathogens, right? Very hard for pathogens to penetrate these layers on their own, right? They usually have to wait for some type of abrasion or injury to enter in. That's why patients that have really bad burns, right? We get worried because the pathogens have a way in. If you've known somebody that's had a compound fracture, I hope you don't, or I hope you haven't had one, but that's actually one of the major concerns with that. Not only, blood loss potentially in their bone, but the fact that that bone penetrating the skin is now allowed pathogens a way in, right? When you shed your skin, you remove microorganisms, right? You shed somewhere between 10 and 15 pounds of skin a year, right? Along with a lot of pathogens. And then you have resident cells that actually live within your skin, right? That can phagocytize pathogens. You also have, I'm not going to talk about this quite yet, but you have resident T cells, which are part of the adaptive immune system, that live in your skin and hang out and are ready to fight pathogens. Below that, right, you have the dermal layer, right? And this dermal layer, right, is composed of collagen, like really tough connective tissue, right? And it helps, again, provides another layer of protection potentially for pathogens penetrating further down. The problem is, though, and it doesn't really have this up here, once you get past the epidermal layer, which is non-vascular, and you enter the dermal layer, which does have a vascular system, right, that might allow access, right, to other areas of the body. Got it? Alyssa? No, the real Alyssa. Good? Sarah, you good over there? All right. So here's, one of the things that I already mentioned, right? But skin, right, has things that are secreted onto it, right? I already mentioned it's already salty, right? So that inhibits the growth of pathogens. You have antimicrobial peptides that can be secreted onto the skin, right? One of those actually being lysozyme. Who discovered lysozyme? Who discovered penicillin? Okay. So you look that up for Monday, Michael, and then you tell me, deal? All right. I'm going after you hard, right? You also have sebum, right, secreted by your sebaceous glands, which are associated with hair follicles, right? This is a fatty, oily substance that actually helps keep your skin nice and pliable so you don't turn into beef jerky, right? And that prevents breaks and tears, right? Those of you that maybe had bad acne, right, when you were younger, these sebaceous glands actually get blocked, and it creates a nice environment for bacteria, and it causes inflammation and swelling, and that's one of the culprits in acne, right? Your skin also has a lower pH on it than other areas of your body, and this low pH, right, prevents the growth of certain bacteria. And we'll talk about that a little bit more further along in the semester, all right? But one of the things I hope you can appreciate from this is that if you look at all these kind of different things that your skin can do, it is a great defense mechanism, right, against pathogens. All right. So, mucous membranes. This is typically the most common site of pathogen invasion, right? And again, all body cavities that are open, right, to the environment. So that's going to be, bless you, right? Your respiratory tract, right? Your digestive tract and your urogenital tract. Okay? Those are all mucous membranes. Those are all open to the outside environment. In fact, the alimentary canal, part of your digestive system, is considered a completely open system, right, from your mouth down to your anus, right? That technically is an open tube, even though we don't think of it like that way, right? That is an important mucous membrane that pathogens can take advantage of.
You said digestive, urogenital, and then what else?
Respiratory. You're welcome, right? So there's mucus, right, that covers epithelial cells, right? These epithelial cells are tightly packed together. How many of you have had ANP1? Yeah? All right, cool. So you probably remember that epithelial cells are held together by things called tight junction proteins. All right, so if you have two cells side by side, right, they're linked really, really tightly. The idea being they're not going to let things easily pass between them. Right, so you have mucus, which is nice and sticky, epithelial cells that are tightly packed together, right? And then kind of like your skin, you are actually continually shedding these epithelial cells, right? And you're getting rid of this mucus, right? This mucus is moved by ciliated cells. Well, specifically ciliated cells in your respiratory tract, right? They're creating this little wave-like motion, causes mucus to move up. You cough, you either spit it out or you swallow it, right? When you spit it out, you remove the pathogen. When you swallow it, the pathogens get exposed to stomach acid. And so it really is kind of a cool system. Hanging out below the epithelial cells, you have dendritic cells. This is a really cool system in the GI tract. Dendritic cells will actually extend their pseudopodia, right? So they're little dendritic cell arms through the tight junction proteins, and they can grab pathogens and they can sample what's in the lumen. Interestingly enough, pathogens have also learned to take advantage of this. They purposely get caught to gain entry sometimes past this epithelial cell layer. And then these chemicals that defend against pathogens, what's really cool is these epithelial cells will secrete antimicrobial peptides that will become embedded within the mucous membrane, right, that mucus layer, and it's basically kind of like a minefield. So pathogens get trapped in the mucus, and then they interact with these antimicrobial peptides, and then you remove the pathogenic threat. All right. So, and that's why I had goblet cells, right, are one of the main cells that produces mucus. Okay. So this is just a picture, right? This is showing you skin, and so you can see all these cells stacked tightly on top of one another and below that connective tissue, that dermal layer. It's a pretty thick barrier. Here you see a mucous membrane. This is actually a fluorescent image. Here you can see intestinal villi. Here you can see bacteria. And this black space, right, is not just a void, right? This is actually a mucus layer, and you can see it's preventing the interaction of the majority of these pathogens, all right? Truth be told, some pathogens interacting with the epithelial cells is a normal function, right? But again, that's a very select number of bacteria, the type of bacteria. So you want to keep the majority of these away. All right? These are some of the antimicrobial barriers that we talked about. So we talked about salt, we talked about lysozyme. Peroxidase can be secreted. This is actually an image from a paper in Nature. It was one of the first papers that was given to me to read in grad school when I joined my one lab. These are cells called Paneth cells. They exist in the crypts of your intestinal villi, and they secrete a bunch of these antimicrobial peptides that then kind of float up and help regulate the bacterial populations that are present in your gut. All right. You also have secreted within a host lactoferrin and transferrin. Does anyone know what lactoferrin does, or can you tell from the name, or transferrin, what they're associated with? Iron, right? And so pathogens love iron, right? Iron is important for a lot of enzymatic functions that are involved in things like nucleic acid synthesis. And so they want it just as bad as we want it. And so these are a couple of molecules that we produce that allow us to sequester iron from pathogens. Okay? You also have defensins, which are kind of like what these are. These are basically little cationic peptides that punch holes in the cell walls of bacteria. We can regulate pH. We also have microbes, which we'll talk about here in just a little bit, that are resident, part of our normal flora. And these help protect us. And then we have complement protein, which we'll talk about a little bit later. All of these helping with that kind of first line of defense. So this is just a diagram, from your textbook, showing you some of the things that your body does to get rid of microbes or pathogens, and you see a lot of it is just kind of normal everyday functions, right? Saliva, stomach acid, right? Bile. Usually you're not eliminating pathogens by vomiting on a regular basis, right? But it does help, right? Every time, right, you go to the bathroom, right, either way, you are potentially removing pathogens from the system, right? So our body is very efficient at getting rid of these. Right? You see other things here, right? Just continuation of that table. So let's talk really briefly about the microbiome. Again, this is your normal flora, and microbiome, the one that gets talked about the most is going to be your gut microbiome, but you have a skin microbiome, you have a urogenital microbiome, you have a respiratory microbiome, right? Pretty much everywhere on your body, you have some type of resident bacterial population that is important to you, right? And the most basic way to look at this is if they are living there, if your normal flora is living there, it prevents, right, somebody bad from moving in. Right? Sometimes it's nothing more than just a space occupation, right? So if someday when Alanis becomes rich and successful, and she decides that she doesn't like people, so she buys a house, and then she buys all the property around her house, so that no one can build in that area, right? So she's occupied that space. But you like people, right? Sometimes, notice. All right. So when space is being occupied, right, consumption of nutrients. So, in some cases, there's not much of a difference between what bad pathogens want and what good microbes want. And by eating it before they can get to it, right, you take their nutrient source, right? Creating an environment that's unfavorable to other microorganisms. An example of this would be lactobacillus, right, are a common bacteria in a lot of flora, and they produce acid, right? And when they produce acid, they produce, right, or that causes a shift in pH. And again, that shift in pH can make an unfavorable environment for a lot of organisms. Prevent pathogens from attaching to the host. Again, if you're occupying the space that's close to the epithelia, pathogens can't get in, they can't interact there, right? This is really kind of interesting, is that your normal gut flora is actually really important for activating and educating your immune system. And so research has shown and continues to show that if you don't have a normal flora when you're young, you have an increased likelihood of having autoimmune disease and allergies, right? And that's because your flora is important for educating. Bacteria themselves can produce toxins that kill other bacteria. So they're great at fighting each other off, right? Again, if you're competing for an environment, you probably have a way to protect yourself. And they also are really important, right, for acquiring nutrients from the environment, right? And so certain aspects of bacteria actually lead to the generation of vitamins that we consider essential. All right? So your microbiome is really important. Talked about this already with mucous membranes. Mentioned this before, right? But your blood, right, can be important. Clotting factors can actually prevent pathogens from gaining entry through a wound. Truth be told, some bacteria have developed ways to break apart blood clots in order to penetrate and enter the host, but still can be important for keeping a lot of pathogens out. We talked about lactoferrin and transferrin, and there's other molecules that are produced by hosts to hold on to iron, right? And then within the blood, again, we keep on mentioning it, and I promise we'll talk about it, but you have complement proteins that are floating around, and these complement proteins are great at interacting with pathogens when they encounter them, right? So again, there are multiple, right, steps, right, multiple things that pathogens have to pass or get through in order to cause infection. I think it maybe can help you appreciate from a biological standpoint how impressive it is that you even get sick, right? A pathogen has to go through a lot to cause illness. Right? All right. We'll briefly start here. So what we're going to talk about in the innate immune system are going to be these cells right here. Right? So these granulocytes, which include eosinophils, basophils, neutrophils, and these monocytes, which can become macrophages or dendritic cells, right? And we'll kind of hold off on these for a little bit later, although we will mention some of these cells of the adaptive immune system. You can see, not surprising for some of you that have taken biology courses in the past, that we're starting up top with the hematopoietic stem cell, right? The hematopoietic stem cell can go one of two ways, right? It can become a common myeloid progenitor, which leads to the generation of innate immune cells, or it can become a lymphoid progenitor, right, which is going to lead to formation of the cells of the adaptive immune system, right? So we're focusing on that myeloid progenitor, which then becomes the granulocytes and or that monocyte potentially, which can become one of two things. So these are your granulocytes. And as you can see, even on this projector, I know the resolution is not great, but they have granules, right? And these granules often contain antimicrobial peptides. You can see that they are relatively distinct in granule color and nucleus shape. Up top, you have a basophil. Actually, I think this is not the best representative picture of a basophil. Typically, a basophil, the granules are so dark, like a dark blue purple, that you can't even see the nucleus. Note too that these granulocytes are somewhere in the range of at least 2 to 3 times bigger than a red blood cell that you can see in the background. Right? You also see your eosinophil here, red granules, bilobed nucleus, and then your neutrophil down here, which makes up the majority of your circulating white blood cells. And they can have a nucleus that has as many as 5 lobes, right? It's all one nucleus, it's just bent in 2 different directions. Right? Neutrophils mainly are phagocyte. Eosinophils and basophils are releasing compounds such as histamine and heparin, right? So they play a major role in inflammation. And if any of you have allergies, right? These cells are also contributing to some of the unpleasantness that you experience with allergies. So as I mentioned, right, neutrophils, over 50% of your circulating white blood cells. I'll probably talk about this more in just a minute, but they're the first responder. All right, so whenever you have an injury, whenever you have a pathogen, it's going to be neutrophils that are first on the scene. Basophils, right, are releasing histamine. What does histamine do? It's an inflammatory response, right? So you think about all those unpleasant things that you associate with allergies, right? That's actually a good thing when you have a major injury or you have a pathogenic infection, because that means you are bringing cells such as neutrophils to that injury site to help control that infection. Eosinophils are typically involved in parasite defense. So some of you that have gone to fun places in the world, right, and have maybe come back feeling a little ill, have maybe had a parasitic infection. If a doctor were to look at your whole blood cell count and they could look at eosinophils, they'd see an increase there. They also release histaminase to counter the effects of histamine from basophils, right? Your body is incredible at being able to turn off things that it has turned on, right? And we will stop right here because we are out of time. We'll pick up the innate immune system on Monday, see how far we can make it through it. I hope you guys have a great weekend. Please remember your personal reflection paper is due on Monday. There's a Dropbox on Moodle, right? Again, it's a personal reflection paper, so there's a prompt on the microbiology syllabus addendum, so take a look at that. Should not be a killer. Please, do it, and you will be fine. Good? Yes, ma'am. Yeah, I need to make, that's my one thing I didn't get to yesterday in my planner. I'm going to put a sign-up sheet on Moodle, and then you guys can pick the date that you want to go. That'll be kind of spread throughout this semester. All right? Good question. Yes, ma'am. And but you can get on others.